Hi everyone, welcome to AO Tutoring, and today we're going to be going through the Higher Human Biology 2023 Paper 1. Okay, so question number 1. Which row in the table matches the type of cell division that occurs in germline cells with a description of the gametes produced? So we've got a mixture of mitosis and meiosis as our answers, and diploid and haploid. It's always good to highlight keywords in our question that might point us towards an answer. So the first keyword that I'm looking at here is germline cells, okay? And gametes produced. So why are those keywords? Those are keywords because we know that in order for gametes to be produced, they must be produced by the process of meiosis. That's right. So that means that option A and option B are incorrect. And we also know that because we're talking about gametes, gametes are always haploid. Remember, haploid means having 23 chromosomes. Diploid means having 46. So the answer for this question is going to be D. Question number 2, which of the following statements about DNA replication is correct? So we're talking here about fragments and a bit about which strand we're joining these fragments together on. So again, in our options, we've got a mixture of leading strand and lagging strand, a mixture of ligase and DNA polymerase as our options there as well. So just to refresh our memory, this is a little image that kind of shows us the process of DNA replication. Here we can see the leading strand, which replicates continuously. As you can see in this diagram, there's no breaks, there's no stoppages, and there's actually no fragments on the leading strand. So we know, because there's no fragments on the leading strand, we know that leading strand, any option that includes leading strand, has to be incorrect. Because, like we can see in this image, the leading strand has no fragments. So that leaves us to look at the lagging strand. Now, the lagging strand does require DNA polymerase to add those nucleotides on. However, those are just for adding on the nucleotides. When it comes to joining those fragments together, that is the job of our handy dandy friend, DNA ligase. So the answer for this question here is going to be B. The lagging strand is joined together by DNA ligase. Question number 3. PCR was used to amplify a region of DNA. After 5 cycles, 32 copies were present. Calculate the number of additional copies present after 4 further cycles. Now, we've always got to take note whenever they highlight something to us in bold. That means it's something that we've got to pay attention to. So they want us to draw attention to the idea that we have additional copies, which really means we're going to be doing a bit of maths and probably taking away our original number from the answer we get when we do our calculation. So just to remind ourselves, this is an image that kind of talks about the process of PCR. We know that we heat our DNA sample to a certain temperature, we cool it down, and then we heat it up again. It's all about the adding of the primers. Not exactly what this question is asking about. So we're going to look at this part of the diagram. So we can see that after the first cycle of PCR, we start with 1 piece of DNA and we end up with 2. After the second cycle, we end up with 4. Okay. After the third cycle, we end up with 8. 1, 2, 3, 4, 5, 6, 7, 8. So what that means is that there's a calculation we can use when we're calculating it. The number of PCR copies we're going to get is 2 to the power n of how many cycles we've done. So if we do 1 cycle of PCR, we're going to get 2 to the power of 1, which is 2 copies. If we do 3 cycles of PCR, we're going to do 2 to the power of 3, which gives us 8 copies. Now that's helpful because as we're going to look at this question, it's going to help us to have a shortcut in figuring out how many copies we're actually going to get. So for this question here, what we're going to have to do is we're going to say that after 5 cycles, 32 copies were present, which means 2 to the power of 5 is 32, which is great. Now, we're going to calculate the number of additional copies. The thing is, that means we're doing 2 to the power of 9 cycles of PCR. If we do 2 to the power of 9 cycles of PCR, we'll get an answer of 512 copies. Now, it might be really tempting to just jump at this answer, but hold on a second. We're being asked about additional copies. So we need to take that original 512 value and take it away from the 32 we started with. And that gives us 480. And this is the number of additional copies we're going to get after we do 4 more cycles. So the answer here is C. Okay, so question number 4. The list shows the nucleic acid molecules involved in gene expression within a cell. You've You've got RNA, rRNA, mRNA, DNA, and tRNA. And your question is, which of these molecules are required for translation? So we know that protein synthesis happens in two stages: transcription and translation. Remember, that's translation is the part two. So this is the second part of the process after we've already created that mRNA copy and it's left the nucleus. Now, because it's that second part where we've left the nucleus, we know that it's not going to involve DNA because DNA cannot leave the nucleus with that mRNA molecule. Now, rRNA is a type of RNA that we don't talk about very much in biology, but that is one of the RNA types that the ribosome is actually comprised of. So rRNA is actually what the ribosome is made of. mRNA is our messenger RNA, and we know that tRNA is the transfer RNA molecule that helps to bring amino acids based on the anticodon on the tRNA molecule. So because of that, we know it can't be three, so it's not going to be D. It's not just one because we've also got mRNA and tRNA. So therefore, the answer here is going to be C. Question number 5. A student set up a test tube containing 5 centimeters cubed of milk and 1 centimeters cubed of the enzyme trypsin. The milk became clear and the student concluded that the white milk protein had been broken down by trypsin. To show that trypsin caused the milk to become clear, a control tube should contain. So this is an experimental design question asking us about what a control should look like in the context of this specific experiment. So it's really helpful whenever we're doing an enzyme related question to always identify what is your enzyme and what is your substrate. Sometimes as well, they might include an inhibitor, and it's also really important to recognize what in your question is your inhibitor. So there's no inhibitor here. So we're going to first think, right, what's the substrate? Well, we're trying to break down milk. So this is what we're trying to do here. We're trying to break down 5 centimeters cubed of milk. So this milk is going to be our substrate. The enzyme that we're working with is trypsin, and we've got 1 centimeters cubed of our enzyme trypsin. So we've got our substrate and we've got our enzyme. If we want to set up a control for an enzyme question, we want to make sure that the substrate is the same, but we don't yet include the enzyme. We want to make sure that our substrate is the same, but our enzyme, we don't have an active enzyme ingredient there. So let's look through the answers. So we've got A, which is 5 centimeters cubed of milk and 1 centimeters cubed of distilled water. That looks like it can be a contender because we still have our 5 centimeters cubed of our substrate, but we don't have an active enzyme, which is useful for control. But let's look at the other options and see what we think. So 5 centimeters cubed of distilled water and 1 centimeters cubed of trypsin. It's not going to be this because water is not our substrate. So even if we do add trypsin, it's not going to do anything. So that's not the answer. Boiled and cooled milk is also not going to be the answer here because although we're using milk, boiling and cooling milk is not what we're trying to do in the original experiment. So that's not what we're going to do here either. That's going to change the chemical composition of the milk and going to give us a different experiment. So the answer here is going to be A. The adequate control is to use 5 centimeters cubed of milk and 1 centimeter cubed of distilled water. Metabolic pathway used to produce the Question 6. Part of a metabolic pathway used to produce the amino acid isoleucine is shown. I'm going to struggle to pronounce these words. Please don't judge me. So we have threonine and threonine deaminase, ketobutyrate, and isoleucine. And this is kind of showing our metabolic pathway here. We're being told some information here. We're being told that isoleucine is a feedback inhibitor of the enzyme threonine deaminase. I don't know who names these compounds, but it's very hard to say. So those are important words to highlight. Feedback inhibitor of that specific enzyme. So the statements below refer to the substances in this pathway, and our job is to figure out which ones are correct. So let's look at the first statement. Isoleucine binds to the threonine deaminase. Well, we have a feedback inhibitor. What that basically means is that the end product of our metabolic pathway will go back to inhibit an enzyme earlier on in the pathway. So isoleucine will go back to inhibit this enzyme. So number 1 is going to be correct. Threonine deaminase lowers the activation energy required to convert threonine into ketobutyrate. Well, we know that this is an enzyme, and one of the things that enzymes do is they lower the activation energy required for a specific reaction to take place. So we know 2 is correct. Number 3, ketobutyrate is the substrate of isoleucine. Well, actually, ketobutyrate is actually the product that comes before isoleucine. So there's going to be another enzyme, another process here that's going to convert ketobutyrate into isoleucine, but it's not going to be the substrate of isoleucine. So statement number 3 is incorrect. That means then the answer is 1 and 2 only, which is B. Okay, question number 7. The table shows the effect of a competitive inhibitor on an enzyme's activity. So you got concentration of the inhibitor here, which is going up in 0.1. And then you've got enzyme activity, which is a percentage of the total enzyme activity. So at 0 concentration of inhibitor, we've got 100% enzyme activity. The enzymes are working fine. Once we hit 0.40, we get 0 enzyme activity. So from 0.40, we know that's the point of inhibition. Not what the question is asking here, but good to always recognize what's happening with inhibitors. We're being asked to predict the concentration of inhibitor when half of the active sites of the enzyme are occupied by the inhibitor. Well, if half of the active sites are occupied, we're thinking that enzyme activity is going to be roughly 50%. So let's look at our enzyme activity column, and let's look to see where we'd find 50%. 50% would be between these two values here, between 60 and between 40. If we look at the concentration of inhibitor, that's 0.10 and 0.20. So we're looking for a value that's slap bang in the middle of these two values, and that's going to be 0.15. So question number 8. An investigation was carried out into the effect of substrate concentration on the rate of a reaction in the presence of a competitive inhibitor. Which of the following would ensure the results were valid? So let's take a second to figure out what we're doing here. So we're doing an experiment to see the effect of substrate concentration on the rate of a reaction in the presence of a competitive inhibitor. So what does that imply? That implies that what we're going to be changing in this experiment is we're going to be changing the substrate concentration. So we're going to be increasing or decreasing the substrate concentration and seeing what that does to the rate of the reaction in the presence of a competitive inhibitor. Okay? So if we wanted to make sure the results were valid here, we have to think about what we're studying. What we're studying is the substrate and the effect the substrate concentration has on the rate of the reaction. Now they're telling us that we're in the presence of a competitive inhibitor, but they're not telling us that we're changing the concentration of this inhibitor or the volume of the inhibitor or anything like that. So let's think about these options. If we repeat the experiment using a non-competitive inhibitor, that's not helpful for us because we're doing this in the presence of a competitive inhibitor. If we keep the inhibitor concentration constant, that might be helpful. Yeah, because the reason that's going to be helpful is we want the inhibitor concentration to be constant because that's actually not what we're studying. We're studying the effect of substrate concentration on the rate of the reaction. So the competitive inhibitor concentration is a bit like a constant variable. Let's look at option B, keeping the inhibitor concentration constant. So that might be really helpful for us because the competitive inhibitor concentration is a bit like a constant variable. It's just going to be a constant thing in our experiment. It's not actually what we're studying. What we're studying is the effect of substrate concentration on the rate of the reaction. So let's keep B in the running. So C, repeating the investigation with each concentration of substrate. But that's kind of what we're trying to do anyways. That's not going to change the validity of the results. That's just the experiment that we're doing. So C is what we're going to be doing. It's what we're planning to do. We're planning to change the substrate concentration, but it's not going to ensure the results are valid. And D, keeping the substrate concentration constant. Well, that's not what we're trying to study. We're trying to study the effect of that changing substrate concentration on the rate of the reaction. So that means there, the answer is going to be B. Okay, question number 9. During aerobic respiration in a cell, 1 molecule of glucose yielded 38 molecules of ATP. During respiration without oxygen in the same cell, 1 molecule of glucose yielded 2 molecules of ATP. Calculate the percentage decrease in ATP yield per glucose molecule when this cell carries out respiration without oxygen compared to when it respires aerobically. Right, so this is going to be a bit of a maths question. Oops, if I can draw a straight line. So we're going to be using 38 molecules of ATP and 2 molecules of ATP. And what we're being asked to look at is a percentage decrease. So I think the easiest way sometimes to calculate this is using the little formula here, the change over the original value times 100. So whenever you're being asked to calculate a percentage change, you can always use this formula. The change in the values over the original value times 100 will give you the percentage increase or decrease. So let's think about the change. Now, we've started off with 38 molecules of ATP, and the change between 38 and 2, if you take away that 2, that change is going to be 36. So the change between those two values is 36. The original was 38. We times that by 100. That gives us a funny looking decimal. I think it's 94.7 something percent. And that will round up to 95%, which is option C. Question number 10. The diagram represents some of the processes that occur in a mitochondrion. Which letter represents ATP synthase? So this is a question about the electron transport chain, and this is the end process of respiration that's going to produce our ATP for us. So A here is going to represent a membrane protein. B is also a membrane protein. C is going to represent an electron. We're going to be pumping our hydrogen ions up here, and these electrons are going to be carried along the chain. And these hydrogen ions are then going to come out here and come down through the ATP synthase molecule to produce for us our lovely ATP. So the answer here is going to be D. Question number 11. Which of the following applies to the skeletal muscle fibers that are most useful during a cross-country ski race? So if we're thinking about cross-country ski racing, this is a long-distance endurance kind of activity. So when we have long-distance endurance activities, we need lots of mitochondria to produce lots of energy for that whole duration. So we're going to have a high amount of mitochondria. So automatically A and C are out the running. Now, if we're going to be running for this huge period of time, we have loads of mitochondria. We're going to need a good blood supply to allow enough oxygen to get to that muscle tissue. So we're going to require a large blood supply, which is great because that's what we've got over here as well. Now, if we have this large blood supply and these huge number of mitochondria, we only ever need loads of mitochondria if we're doing aerobic respiration. So the answer here is going to be D, because we're going to need those mitochondria to help us respire aerobically and produce all the ATP we need for this cross-country ski race. Okay, moving on to Unit 2 with question number 12. Which hormone found in contraceptive pills causes thickening of the cervical mucus? So, because we're talking about contraceptive pills, we're not going to be talking about FSH or LH. These are our pituitary hormones, and you might find those in infertility treatments that help to stimulate ovulation and things like that. But when it comes to contraception, we don't typically use these hormones. So we're really between estrogen and progesterone. Now, thickening of the cervical mucus is something that progesterone is going to do. Estrogen is going to help thin out that cervical mucus and to allow sperm to enter during that follicular phase. And progesterone, which is the dominant hormone in the luteal phase, is going to cause thickening of that cervical mucus. So the answer here is going to be progesterone. Okay, question number 13. Which letter in the diagram indicates the site of testosterone production? So let's identify what these different structures are. C, that's going to be the bladder. That's where we store our pee. B, this is our seminal vesicle. And A is the prostate. So these two structures are going to help us provide certain nutrients into the semen to help the sperm swim and survive, and lots of enzymes and sugars and things to help them survive their journey to the egg. And we know that testosterone is produced in the testes by those interstitial cells. So the answer here is going to be D. Okay, question number 14. Cystic fibrosis is caused by a recessive allele. The diagram shows the inheritance of cystic fibrosis in 3 generations of a family. Which individual confirms that cystic fibrosis is not a sex-linked condition? Now, a lot of people find these pedigree charts challenging and difficult to look at. Let's try and break down what we're looking at and make it a bit easier to digest. So we're being asked a question: is this a sex-linked condition? We know the answer is no, but how do we know it's not a sex-linked condition? So generally, with sex linkage, the dads are going to pass to the daughters, and the mothers will pass on to their sons. Okay? The reason that is because we know the male chromosome, X, Y. The X chromosome is the only one that can carry on the allele. Let's make the allele for this question F. So if it's a recessive allele, the father here has a potential, if it was a sex-linked condition, would have the genotype X, small f, Y. And so in order for this man with this specific genotype to pass it on to his son, it means that his partner here must also have a recessive allele, meaning that she might be X big F, X small f, and he would be X big small f Y. Sorry. So the recessive alleles here, recessive alleles here. So we know that fathers pass it on to daughters, mothers pass on to sons. So it still could be potentially that the mother might pass it on to the son. So C could be the answer if you think about it without looking at the next generation. C might be an answer that you're thinking of, but don't rush these questions too quickly because we've got another generation to look at. So if we look at this other generation here and we're trying to think, right, what's their genotype going to be? We know this guy, if he's affected, his genotype is going to have to be X little f Y. And this girl here is going to have to be X little f, X little f. Now, she has had children with an unaffected male. So if you're an unaffected male, your genotype is going to be X big F Y, which means that the father will pass on the Y chromosome to their son, and the mother will pass on the recessive allele to their son as well, which makes sense. B could be the person that has that, B could have that specific genotype as well. But if we look at person A, the father will pass down X big F to the daughter, and the mother will pass down X little f, meaning this person's genotype should be X big F, X little f, which means they should not be affected by the disease. But this diagram is showing us that they are affected by the disease. So because of this person here, person A, who is a female, who is affected, but her father does not have the disease, and her mother does carry the recessive allele, she should be X big F, X little f, and therefore should be unaffected, protected by that dominant allele, but she's not. She does have the disease. So this person, because this person is affected, person A shows that this whole thing of sex linkage can't apply to this question specifically because person A is affected and the genotypes show that they shouldn't be. So the answer for this question is going to be person A. I hope that kind of makes sense. Sometimes these pedigree questions can be challenging. If you'd like me to make a video on pedigree charts and how to interpret them, you can comment down below and we can look at that in a further video. Okay, question 15. The following steps are involved in the process of IVF. So you've got all those steps there. I'll give you a second to read those. You can pause the video and read the steps. Which of the following sequences shows the order in which these events take place? So when I think when we're doing these questions, it's always important to look at the first letter to make sure if that makes sense for the process we're looking at. So here, our first kind of numbers, I said letters, but I meant numbers, apologies. So the first numbers that we're looking at are 4 and 2. So we've got 4, 2, 4, 2. If we look at the options here, 4 is eggs are surgically removed, and 2 is woman is given drugs to stimulate ovulation. So automatically, you can't surgically remove eggs that have not yet been stimulated. So anything beginning with a 4 is automatically out. So we're between 2. We're between 2 and we're between B and D. If we look at B and D, their processes are very similar. 2, 4, 3, 6, 2, 4, 3, 6. The only difference is B is telling us step 1 happens before step 5, and D is telling us that step 5 happens before step 1. So let's look at step 5 and step 1. So step 5 is that embryos are implanted. Step 1 is zygotes are incubated. So we have to incubate the zygote before we can implant it. So 1 has to come before 5. Therefore, the answer is going to be B, which means the process is woman is given drugs to stimulate ovulation. Eggs are then surgically removed. Eggs are mixed with sperm in a culture dish. Fertilization occurs. Zygotes are incubated, and then the embryos are implanted. Question number 16. Measurement of a child's cardiac cycle showed that systole lasted for 0.2 seconds, while diastole lasted for 0.4 seconds. What was the heart rate of the child? So this is quite a common question you get for the whole cardiology, cardio type unit, calculating the cardiac cycle. So what are we needing to look at here? So this is a systole and this is the diastole. So this a whole cardiac cycle is systole plus diastole. Both of those together give us one cardiac cycle. So our systole was 0.2 seconds. Our diastole was 0.4 seconds. So the whole cycle was 0.6 seconds, which means we get 1 beat per 0.6 seconds. Now, if we want to ask ourselves, how many beats are we going to get in a minute? Well, let's try and see what we can do to turn this 0.6 into 1 minute. So 1 minute is 60 seconds. So to get this to become a minute, you would times this by 100, which means you're timesing this by 100, which means that you're going to get 100 beats per 60 seconds. So therefore, your answer is C. Question number 17. Peripheral vascular disease most commonly occurs in the arteries to the. So this is a question that's really asking us about what do we know about arterial disease and what arterial disease and what part of the body causes what kind of disease. So arterial disease in the heart causes a heart attack. Arterial disease in the brain causes a stroke. We don't usually get arterial disease in the lung, but you can. Not something that's commonly spoken about. So we'll cross out lung because it's not something that we ever talk about when it comes to arterial disease. And then there's arterial disease in the leg. Now, brain and heart, whenever you get arterial disease in the brain and the heart, we don't call it peripheral vascular disease because it's not in the peripheries. Those are very central parts of our bodies where you can think about it. Peripheral is really referring to something that's kind of far away from the center of your body. And vascular disease, arterial disease in the heart and in the brain, we have names for those. We only really use the phrase peripheral vascular disease when we're talking about vascular disease in the legs. So this is a question that's a bit about wording and the words that we use for things. So peripheral vascular disease is, again, most commonly used to describe vascular disease in the legs. In the brain and the heart, we use different phrases. So question number 18. In a study, the percentage of people aged 55 years and older who used statins was recorded in 1990, 2000, and 2010. And the results are basically shown in this graph. Our job is to figure out which one of these 4 statements is correct. These questions can be a bit time consuming because you really need to look at every single option to figure out which one is the correct option. You need to interrogate all the different possibilities. So let's do that together. Between 2000 and 2010, statin use increased in all age groups. So between 2000 and 2010, so for the 75 plus, that's true. But for the other 2 age groups, the 65 to 74 and the 55 to 64, it actually decreased. So A is not going to be our answer here. Then we've got between 1990 and 2010, statin use in the 75 plus age group tripled. So 75 plus, that's our gray bar, that is at 12% over here. And we look at 2010, if it were to be tripled, it should be at 36%, which is 3 times 12. But we can see here it's well above 40%. So that's actually going to be an incorrect answer as well. And between 1990 and 2000, statin use in the 65 to 74 year age group doubled. So let's look at 1990 and 2000. So 1990, we're at 11 here. And in 2000, we're actually hitting at 22. So that looks like it's going to be true. Let's look at that final option just to make sure we're not missing anything. Between 1990 and 2010, statin use in the 55 to 64 age group decreased. So between 1990 and 2010, they were both the same. They're both about 1%. So it didn't decrease, it actually stayed the same. So the answer here is going to be C. Question number 19. Which row in the table matches the branch of the autonomic nervous system with its effect? So it's always really important when you're thinking of autonomic nervous system to split sympathetic and parasympathetic in your mind. Sympathetic, that's your fight or flight. That's when we're thinking of, adrenaline and heart rate increasing and all the kind of things that are going to keep you more alert. Parasympathetic, that's different. That's our rest and digest. That's making sure we can digest our food and all of those good things there. So if we're going to look at this, we're going to look at the sympathetic thinking. Sympathetic's got to be right, fight or flight. Parasympathetic is going to be rest and digest. Okay. So if that's the case, If we have a sympathetic branch and it increases intestinal secretions, that cannot be correct. The reason is intestinal secretions will increase when you're resting and digesting. B, sympathetic and causing a decrease in your breathing rate. Well, actually, the sympathetic response should be an increase in your breathing rate to help you with oxygen delivery more efficiently when you've encountered some sort of threat, maybe a bear or something like that is chasing you in the woods. You need to have an increased breathing rate so you can get more oxygen delivery. So that's also going to be incorrect. C, parasympathetic. So now we're thinking on the rest and digest side, decreasing intestinal secretions. We would want that to increase intestinal secretions. So that leaves us with the final answer here, which is that the parasympathetic response is going to cause us an increase in peristalsis, which is D. Question number 20. Which of the following is not a method of encoding information into long-term memory? So when we're moving from short-term memory to long-term memory, there are three different methods we can use. We can use rehearsal, elaboration, and organization. If you're studying for your exams, you've probably been doing a lot of those things. The one that stands out here is chunking. Now, chunking is not to help us move from short-term memory to long-term memory, but it's used so that we can hold more information in the short-term memory by splitting it into chunks. So you remember your phone number in 3 chunks usually, instead of in one massive chunk, one massive number. So chunking is to keep more space in the short-term memory by basically chunking up the information that we have, making it easier to remember. But the other 3, those are how we encode from the short-term memory into the long-term memory. So the answer here is going to be A. Okay, question number 21. I'm going to have to scroll so you can see the answers after this. Split brain patients cannot transfer information between the right and left hemisphere of the cerebral cortex because the corpus callosum has been cut during surgery. In a study, a split brain patient was asked to press their forehead against a barrier so that their left eye could only see the left of the barrier and their right eye could only see the right of the barrier. So this kind of image shows the experimental setup. So we've got a specific pattern here on the left side of the screen, another pattern here on the right side of the screen, and there's a barrier there to restrict, the field of vision there. We've got the cut corpus callosum, right cerebral hemisphere, left cerebral hemisphere, and the visual areas of the brain. The patient was asked to look straight ahead, and then 2 patterns were flashed briefly on the screen as shown. Which of the following diagrams represents where the patterns are interpreted in the visual areas of the patient's cortex? Now, this is a tricky kind of question to understand. And again, if you'd like to have another video about this or a deeper explanation, you can put a comment down below. We can go into this in more depth. But essentially, what we need to do to understand this is understand what the normal vision should be like and what's happening when you've cut the corpus callosum. So let's take a look at this diagram I've put on the screen here. So to understand this phenomenon a bit better, I've put this diagram on the screen so we can kind of look at it and see what's happening here. So the key thing to look at for this is where is the visual field, which part of the eye interprets that visual field, and which part of the brain will then interpret that result. So the key thing to note here is that the right visual field will be interpreted by the left side of the brain. You can see this blue here is from the right visual field on the right side of the eye, the left side of the eye into the left part of the brain. The left visual field picked up by the right side of the eyes and interpreted in the right brain. So that is what is going to happen. The right side of the visual field will be interpreted in the left part of the brain because there's some crossover here. The difference between a normal brain and what happens when you cut this corpus callosum here is that you don't get communication between the hemispheres in the brain, okay? Which means the left eye will see the right visual field, but instead of passing on that information to the right side as it should, the left eye is going to see the right visual field and interpret that in the right part of the brain, but there won't be the communication from the left side of the brain to the right side of the brain. So it's not going to really pick up on this left visual field as well. So that means that whatever is in the right visual field, the left eye and left brain will pick up. In the same way, whatever's in the left visual field, the right side of the eyes, and here the right eye, and the right part of the brain is going to pick up that signal and it's going to process it. And we're not going to get the crossover from this part of the brain, which means instead of having this nice integrated picture, kind of talking about these 2 things that we're seeing at once, we're going to see two distinct things with very little crossover in the middle. So if we go back to our question and see how that relates, then if we have this zigzag pattern happening here in the left visual field, that's going to be interpreted on the right part of the brain. And this straight up and down pattern, this line pattern on the right visual field is going to be interpreted in the left part of the brain. Okay, but there won't be a crossover in terms of what the left eye is seeing and the right eye is seeing. They're going to see them as two distinct images. Interesting phenomenon. So the answer here is going to be C, because C is showing us that, I'm trying to get them both in view. Let's see if I can shrink this a wee bit. C is showing us that this stripy up and down pattern is seen in the right visual field, so it's interpreted in the left part of the brain. The cross pattern is seen in the left visual field, so it's interpreted in the right part of the brain. And that's how you answer that question. So question number 22, an individual displayed a hypersensitive response to a foreign substance. Which row on the table identifies the cause of this response and the type of lymphocyte involved? So whenever we're talking about hypersensitive response to a foreign substance, we're really talking about an allergy. That's why when the pollen count is high, we all start sniffling and your throat gets itchy, or if there's peanuts in your food, people have peanut allergy, you get an allergy to a foreign substance. Okay, so that tells us that the answer cannot be A or C, because that's talking about autoimmune disease and we're thinking about an allergy. Now, the important thing to understand is that allergies are always B cell mediated. Okay, it's autoimmune disease that would be T cell mediated, but since we're talking about an allergy, the answer here is going to be B. Question number 23. During an epidemic, the R number refers to the number of cases of a disease that are directly caused by contact with one infected individual. During an epidemic, an individual becomes infected when the R number is 2. This means that during the first level of transmission, 2 further people will be infected, who will then infect 2 people, and then so on and so forth. Predict the total number of people infected after 4 levels of transmission. So I've already drawn out this little diagram here of this person here who's gone on to infect all these different people. So if we have this as the first level of transmission, this infected person infects these 2 infected people. Second level would be these people infecting these people. Then we have the third level of transmission, and then up here we have the fourth level of transmission up here. If we count up all these little dots, and you can draw this diagram yourself, you basically just draw 1, and then every one person infects 2 other people. If you count up everyone here, you get 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31 people. That's a super spreader. Okay, last 2 questions here. Question 24, the diagram shows a lymphocyte releasing proteins it has produced. Which row in the table identifies the type of lymphocyte and the proteins produced? So this question, we can, different ways to think about it. I think it's easiest to do by process of elimination. So here we can see the proteins that are produced. These are Y-shaped proteins. Now, when we see a lymphocyte producing Y-shaped proteins, we can automatically think, oh, that's going to be an antibody, because antibodies are Y-shaped proteins. They kind of have this general kind of shape like this. So they're kind of Y-shaped. And so whenever we see a Y-shaped protein, we can always think, right, okay, that's going to be an antibody. The other way we can think about it or get to the idea that we're dealing with antibodies is that antigens are surface proteins that help to identify a cell. So cells are not going to release an antigen unless it's an antigen presenting cell or it's a phagocyte that's digested a cell and is expelling its contents. So because of these Y-shaped proteins, we could think, right, antibody, or because we're thinking antibody, antigen, we don't really produce antigens. Antigens sit on the surface. You'll get to the idea that we're dealing with antibodies. So anything that says antigen, you can get rid of. What type of lymphocyte produces antibodies? Well, there's only one type of lymphocyte that's going to produce antibodies, and that's going to be a B lymphocyte. So the answer here is A. And finally, the last question. Question 25, the list contains statements about the factors considered during the vaccination of a population. So the type of disease, population density, effectiveness of vaccine, and the percentage of non-immune individuals. Which of the statements refers to factors that can affect the herd immunity threshold? Okay, so When you're thinking about herd immunity, what are the things that are important? The type of disease, that's really important because the type of disease will be impacting, how it transmits, the effect of the disease on the individual, the infectiousness of the disease, how contagious that person is, and all those different kind of things. And that's going to depend on the disease. Number two, population density. What is the likelihood you're going to run into somebody who has the disease? How densely populated is that specific place? And then, therefore, how easy is it to transmit or to spread that specific disease? Number three, if we're thinking about vaccination and we're thinking about herd immunity, we need to think about how effective is that vaccine? Because if the vaccine is not effective, then that's going to impact the vaccination program and then therefore impact herd immunity threshold because the vaccine is not particularly very good. So, statement number four, the percentage of non immune individuals. So, this statement is going to be incorrect. So, let's think about why this is incorrect. So, let's think about the definition of herd immunity. So, herd immunity is the resistance to the spread of an infectious disease within a certain population that is based on pre existing immunity of the people in that population, and is usually based on a high proportion of individuals who are either immune through previous infection or immune by vaccination. So when we're thinking about herd immunity, we're thinking about people who are immune, not the people who are non-immune. So because we're not thinking of people who are non-immune, we're thinking about people who are immune and therefore protect everybody else, then statement number four is going to be incorrect. We know statement number one is correct, statement number two is correct, statement number three is correct. So therefore the answer for this one is going to be C. So thank you guys for watching this video going through the 2023 Higher Human Biology paper 1. And if you like this video, please like and subscribe, and comment down below if there's anything you didn't understand or if there's any other topics you'd like to kind of have a further explanation on. But I hope you have a wonderful day.